Today we're gonna talk about DNA replication. First, let's talk about DNA helicase. DNA helicase is an enzyme that unzips the DNA. DNA is normally double-stranded, so it must be opened before replication can happen. Helicase break the hydrogen bond between the base pairs, allowing the two DNA strands to separate and replication to begin. Next, DNA structure. DNA has a double-stranded structure called a double helix. During DNA replication, these two strands separate. When they split apart, they form a Y-shaped structure called the replication fork. Each separate strand is then used as a template to make a new strand. Next, DNA bonds. The two DNA strands are held together by a hydrogen bond between the base. Adenine pairs with thymine using 2 hydrogen bonds. Guanine pairs with cytosine using 3 hydrogen bonds. These hydrogen bonds are what keep the DNA strand together, and they are the bond that helicase break to open the DNA. Next, DNA polymerase. The enzyme that builds a new strand. DNA polymerase adds a new nucleotide one by one to form a new strand. However, it can only add nucleotide to the 3 end, so DNA synthesis always happens in the 5 to 3 direction. DNA primase and DNA ligase. DNA polymerase cannot start replication on its own. That's why we need DNA primase. DNA primase creates a short RNA primer which provides a starting point for DNA polymerase to begin building the new DNA strand. Now let's look up DNA ligase. DNA ligase is an enzyme that joins DNA fragments together. On the lagging strand, DNA is made in a short piece called Okazaki fragment, and DNA ligase fills the gap between them. This is a DNA primase, and this is a DNA ligase. Direction of DNA strand synthesis. Both the new DNA strands are synthesized in the 5 to 3 direction. However, because the original DNA strand run in opposite direction, the two new strands are made differently. The leading strand is synthesized continuously. The lagging strand is synthesized the fragment called Okazaki fragment. Finally, relationship between DNA and chromosome. A chromosome is a long DNA molecule that is tightly coiled and wrapped around protein called histone. DNA contains the genetic information, and a chromosome is the packaged form of DNA which allows it to be stored efficiently and separated correctly during the cell division. That's all for today. Thank you.